So this lesson is going to be on enzymes, and the reason why we're going to talk about enzymes is because we are starting a unit called biochemistry. And if we look at the word biochemistry, we can see the prefix bio, which means living, and in chemistry, which is referring to all the chemical processes that occur in a living organism to keep them alive, like aerobic respiration, production of hormones, lots of different things. So, and enzymes play a huge part in biochemistry because they are organic catalysts. Well, first let's take a look at organic and see what that is referring to. So, in order to be organic, it has to have carbon and hydrogen together in the same molecule. So, if we take a look at glucose, C6H12O6. Okay, this is organic because it contains carbon and hydrogen. So check, that's organic. What about water? H2O. A lot of people think water, we need it to live, has to be organic, but take a look. It only has hydrogen. All right, it's not organic because it's lacking the carbon. What about this one? HCl. Organic or inorganic? Organic, inorganic. It is inorganic because whenever you have a capital with a lowercase, they go together. This carbon isn't referring to, this C is not referring to carbon, but it's referring to chlorine. So therefore, this molecule only has hydrogen. So it is inorganic. Okay, so enzymes are organic and they are a catalyst. So a catalyst is something that speeds up a chemical reaction. And one that comes to mind is, chemical reaction is hydrogen peroxide, H2O2. If you look at this, right, it looks very similar, right? It looks like water, H2O. Hydrogen peroxide will eventually change to water, but it'll take, let's see, maybe about 1,000 years or so for it to go from hydrogen peroxide to water. But we do have enzymes that will make that happen much faster because hydrogen peroxide is a waste product in some metabolic activities that occur in our bodies, and our cells need to get rid of that because I'm sure we've all used hydrogen peroxide before, putting it on a cut. It's designed to kill off the bacteria, so it's toxic. So our cells, as fast as they produce it, they need to get rid of it. So they have enzymes that will get rid of that extra oxygen here to create water, and it happens really fast. And I have an experiment at the end of this video that will show you just how fast it is. Okay. So before we can talk about enzymes, we need to know that they are made up of proteins, and proteins are made up of amino acids. Okay, amino acids are known as the building blocks of protein. Okay, so you need to know that expression, building blocks of protein. I guess the reason why they call it building blocks is because amino acids, you put a whole bunch of them together and it makes the protein, just as you would if you had maybe when you were younger and you had blocks to play with, you put them together to create different structures, right? So amino acids are put together to create many different structures of protein. And pay close attention to this. The order of the amino acids are what determine the function of the protein. So if we take a look at this picture here, we can see that it's this is an enzyme called catalase. And this green ribbon is representing the amino acid sequences, whatever they might be. And this particular sequence of amino acids produces this odd shape. You get lots of bends and curves, loops and curls. If you were to change just one amino acid, you could change maybe you change this loop into something else, and you don't have catalase anymore. You would have something else. So the order of the amino acids are very important. Let's take a look at insulin, right? Let's say this was insulin. If somebody was diabetic, their cells are not producing insulin, so there could be amino acid sequence that's incorrect. So instead of having this loop here, maybe it goes straight and the insulin's not going to function. So the shape of the protein is very important to its function, which leads me to this part, the lock and key. This gray structure here is representing the enzyme, whatever it might be. This green structure here is known as the substrate. The substrate is the molecule the enzyme is working on. And if you look, the enzyme has this part right here, which is known as the active site. And if we look, we could see that the active site matches the substrate. Okay, it's specific. If we look at it, and that's where the name lock and key comes from, because a key, I should say a lock, has a specific key that will make it function, correct? So here, enzymes have a particular substrate that will fit into its active site, and it'll work. If you put in a different substrate, it won't work. Let's try to make it a little bit more specific. So let's say this enzyme is lactase. And this substrate here would be lactose. You may have heard of this before. You may have heard some people being lactose intolerant. What that means is their digestive system can't break down lactose. Lactose is a sugar. It's a disaccharide, which means it's made up of two simpler sugars. One is galactose, and the other sugar, let's say this one here, would be glucose. So you put the two sugars together, and it creates a disaccharide called lactose, and it's most commonly found in milk. So people that are lactose intolerant have a hard time drinking milk because they don't produce the enzyme lactase. So lactase will bond with lactose at the active site. There will be a chemical process that takes place, and it separates the two sugars that make up lactose. What if I had maltose, which looks like this? Would this fit in this active site? Hopefully you said no, right? You can see right away that this shape does not fit. You would need a different enzyme for this particular maltose, for this particular substrate. All right, there's one enzyme for one substrate. And that's going to bring me to our experiment. So here in this flask, you can see I have a liquid. I put some blue food coloring in there just so you could see it a little better. I put some soap and hydrogen peroxide. So hydrogen peroxide, soap, and food coloring. In this beaker here, I have yeast, because yeast will have an enzyme in there that's going to take this hydrogen peroxide and speed up the rate at which oxygen is released. So let's watch the video. Let's try that again. Let's watch the video. Hold your applause to the end, please. So here I'm putting some water into the beaker with the yeast. Swirl it around a little bit, and then I'm going to pour it into the flask, and let's watch what happens. Okay, you can see all the oxygen immediately being released. It's the soap and oxygen is what's creating the bubble, so it's creating this foam that's coming off. Okay, so it happens really fast, and I don't know if I mentioned this before, but the enzyme involved in this could react with 1 million or over 1 million hydrogen peroxide molecules each second, and it's pretty evident in this reaction how fast it takes place. Okay, so what I need you to do is I want you to find out what enzyme in yeast is responsible for this reaction. What enzyme is responsible for that release of the oxygen? How can we test that oxygen was released and caused the production of the foam? So what could we do to see that it was the oxygen? And are there any changes we could do to this experiment to alter the rate of the enzyme action? And I want you to design an experiment. So I'm going to give you a hint. The answer to this is yes. And you have to you have to figure out what changes we can do. Okay, so I don't want anyone coming in saying no. Okay, so it can be changed. I want to know how it can be changed. I want you to design an experiment. I want to know the materials that you would use and your procedures. I don't want you to do it because when you come in class, we're gonna put you into groups and you're gonna take a look at your experiments. You're gonna fine tune it, and then we're gonna do it as a class. So that's your homework, and good luck.